All righty. All right, you guys are awesome. For the most part, you all come on time. Another class is always lagging behind, but you guys are pretty good about being in within the first 5 minutes. I appreciate that. Okay. All right. I'm not going to show this whole video because, like I said, some of you get it. Actually, probably which one would be better? Let's look at that one instead because that's where our issue is. How to read a codon chart. All right, hold on. Okay, I forgot to add the volume on this. So let me go back and do that. All right, so again, today we're just mostly interested in getting you to make sure that your worksheets are done. And I want to, like I said, this class is not struggling so much with codons as the other classes, but being as it's Wednesday and it's kind of a review day, that's what we're going to do. All right, so before I show this, what how we got here is we're trying to make proteins, and we make proteins because DNA tells us to. The DNA is our master set of instructions. Question 1 on the worksheet that asks you, what is the relationship between DNA, the codon, and the protein? DNA's job is to try to tell us how to make proteins. It does that by creating a series of nucleic nitrogenous bases in sequence, and in the sequence of dictating what the protein should look like. The messenger RNA reads it in sets of 3, and those sets of 3 are called a codon. So every 3 nitrogenous bases make 1 codon for 1 amino acid. Okay, so this chart is saying, let's take that codon, those 3 letters, and figure out which amino acid the DNA wants us to find. Okay, so then I'm going to let the video play. So that's where we are. We're up to the chart where we got 3 bases, and you have a whole chain of bases lined up. But when it comes to reading it, it's going to read it 3 at a time. So we're going to take each set of 3 and we're going to compare it to the chart. And I'm letting someone in. And then we're going to figure out what amino acid it wants us to put on our chain. And then they're going to hook up together to make a macromolecule or a polypeptide. Okay. All right. So the whole point of this is to make a protein.
Additional help to read a codon chart. And you've come to the right place. We're going to assume you already have the background from our DNA versus RNA video and our protein synthesis video. If not, you might want to check those out first, or otherwise jumping right into this isn't going to make much sense.
Hopefully it's loud enough for you.
Oh,
it's
and
kind
in
of low for
addition,
me.
we have a video companion handout to this video, which can be helpful. Expand this video's details for the link, as you may want to download it first. So if you had this DNA template here, you'd have this mRNA built. I know this because of how RNA bases pair with DNA bases.
All right, whoops. I did want to stop it there because that's our first step. Okay, the DNA, which is the purple one, is going to need to copy itself. and it copies itself because the DNA never leaves the nucleus. But protein synthesis happens in the ribosome, so that message has to get to the ribosome. So it makes a copy of itself. But you need to also have a note to self is that T always bonds with A and C always bonds with G. But in RNA, we don't have T's, we have U's instead. So now every time I see an A on the DNA, if I am making RNA, I'm going to have a U. Okay, so notice on this orange one, there are no T's on there. Every time you have an A on the DNA strand, you're going to have a U. However, every time you have a T on the DNA strand, you will get an A. So RNA does have A's, but it doesn't have T's. So that's what you have to make sure that you understand those. We call these complementary pairs. They match each other. They complement. They are one's on one side, one's on the other side.
Notice how these mRNA bases are written here in groups of 3? Each is a codon. Bases are read in 3s, so a codon is how these bases will be read. In this example, there are a total of 5 codons, 15 individual RNA bases. By reading a codon chart, you can determine the amino acids that will be brought in by the tRNAs. You can determine the sequence of amino acids that make up a protein. And if you're wondering why this matters, understanding how amino acids are coded for helps us understand human traits and how changes in amino acids have the potential to alter those traits. Not just for humans either, but all living organisms. The codons and their corresponding amino acids have been discovered by scientists. There are many ways you can represent the information in a chart. The most common ways are the rectangular codon chart and the circular codon chart. Let's get started. The first codon, remember codons have 3 bases and codons are on the mRNA, is AUG. That means the tRNA anticodon will be UAC, and we know that because of the base pairing rules. Now, which amino acid will that tRNA carry? Time to use the codon chart. We'll use a rectangular one, and remember, it's the mRNA codon that we look at, not the tRNA anticodon. So notice on the left side of the chart, it is for the first base. The top is for the second base, and the right side is for the third base. We're looking at the codon AUG. We go in order. First base letter, A. We isolate this row here and only this row. Second base letter, U. We isolate this column here and only this column. We've narrowed it down to this area here where they intersect. Third base letter, G. Here. The codon AUG codes for the amino acid methionine. Fun fact, methionine is commonly the starting amino acid for many polypeptide chains, as AUG is a start codon. Next codon is CCA. That means the anticodon for the tRNA will be GGU. Now, what amino acid will that tRNA carry? Time to use the codon chart. This time, we're going to ask you to pause the video while you try to determine which amino acid this tRNA would be carrying. Again, don't forget to use the mRNA codon, not the tRNA anticodon. So in slow motion, let's go through this chart.
All right, so that's an important point. We are looking at what is the messenger RNA saying. So that's the codon that we want. We're not what the DNA said, because technically when you look at the transfer RNA, the anticodon is actually the way it looked on the DNA, with the exception of U and T. But we are reading what is on the messenger RNA.
First base letter, C. That narrows down this row. Second letter, C. That narrows down this column. And therefore, this area here where they meet. Third letter, A. The amino acid proline. All right, the third codon, GUC. This time we need you to determine the tRNA anticodon and also the amino acid that tRNA would be carrying. Pause the video to determine those. The anticodon, based on the base pairing rules, would be CAG. But we used the mRNA codon for this codon chart, and when you used it, showing this in slow motion here, you should have gotten the amino acid valine. Now, with those 3 codons, we used a rectangular codon chart. We mentioned that there are other ways to represent this information, and the circular chart is common to find as well. The circular codon chart typically works by starting on the inside and moving outward. So this 4th codon we have here is UUC. That means the tRNA anticodon would be AAG. But again, we're going to use the mRNA codon in the chart. So we start with the center, first letter U, which isolates to this area. Then we move out 1 to the second letter, which isolates further, U. Finally, we move 1 more time to the C. This is the amino acid phenylalanine. Your turn now to use the circular codon chart with the last codon, UAA. What does the mRNA codon UAA code for? In slow motion here, you can see we get stop. This does not code for an amino acid. Instead, it generally is at the end of a sequence for a polypeptide. And, well, this signals the end of reading the codon chart for this little chain of amino acids anyway. In reality, polypeptides tend to have many...
Okay, I wanted to go back and look at that. All right. So again, you have this long, you're making a protein, you have this long chain of information. You have one that says start. So it kind of starts in the middle of the conveyor belt, so to speak, and it reads to bring all these, and this is short, when proteins have much longer chains of amino acids than this, but eventually when that particular amino acid is, that protein is finished, all the instructions for that protein are finished, the last one is a stop. Methionine is always the start, the last one, and I think there's 3 different ways that you can have a stop codon. So it's not going to bring another amino acid, it's just going to tell it stop looking for amino acids. Okay?
Of amino acids anyway. In reality, polypeptides tend to have many more amino acids than this, and proteins tend to be made of 1 or more of these polypeptide chains. We hope this was helpful, but before we go, we want to mention just a few pitfalls when reading codon charts. Pitfall number 1, not using the mRNA codon. Unless it specifies otherwise, the mRNA codon is what you want to use for the codon chart. Some students will accidentally try to use the tRNA anticodon. Some students will try to use the original DNA template. Now, while there are charts that exist for the tRNA anticodon, or DNA, unless it specifically says otherwise, you should assume it's for the mRNA. Pitfall number 2, getting stuck on the standard rectangular chart. I've noticed that students tend to have more challenges with the rectangular one. We suggest going slowly, and even using a highlighter if you're able to. That way you can see where the first base isolates the row, the second base isolates the column, and the third base finishes in isolating the amino acid. Remember to go carefully in order. Pitfall number 3, not practicing. You gotta practice, that's why this video has a video companion. So let's do some more practice by going backwards. So using this circular codon chart, can you tell me the 2 codons that code for lysine?
Okay, I'm going to pause it there and say we have our own practice chart to work at. So we are going to, and I'm going to make sure that this is in the playlist so that you can pull this up if you need to read it again. All right, but what I want you to look at are the worksheets that we were, so you had a worksheet 1 with the protein synthesis code. And let me do it this way, because if I do it the other way, it's going to pull up the grade book, and I don't want it to do that. So slow. Actually, if I did it in student view, that would have been better. Actually, let me do that. That's faster. All right. So again, what I need for you to do today is make sure that you are finished with the worksheets. I am going to kind of quickly go through these because some of you still have these outstanding, so they need to be done today. All right, so we're going to look at protein synthesis and coding. So that was the first worksheet that you had to do. And again, that's basically what she was going through. So on the first one, you had to translate these codons using one of the charts, and you are to write the amino acid on the line. The second one, they gave you the amino acid and you had to take for example, the word proline, you had to come up to the chart, kind of just scan through and see where you saw P, and then they highlighted the first three initials because that's what's on these charts. But like you saw in the video, most charts might have, if it's a bigger chart, it'll have the word written out in full. But PRO on this chart has 4 different codons. So there's 4 different ways that we can code for that one amino acid. So you were supposed to write all 4 of those on the line. I think 1 or 2 of you, I sent it back because you might have written just 1 or 2. But there's 4 ways to get that particular amino acid. And then there's 2 of them that only have 2 different ways. I think it was the histidine has 2 ways to write that. Serine has 2 ways. Arginine has 2 ways. Glycine had 4 ways, and there was another one of your choices. Valine had 4 different ways. So that was the answer that you were looking for on the chart. The valine, the arginine, the phenylalanine, the pH, that has 2. So you were supposed to write all the different codons that would code for that amino acid. That's for number 2. All right. So this one we talked about what is the codon for the start? What is the codon? And there are 3 different codons that's for the stop. So you need to put those codons there. My worksheet is squished it up. So this actually ACT should be on the end so that it reads like it's a long chain of DNA. Then you were supposed to create the complementary RNA strand. And based on your messenger RNA, like she said in the video, you are to go use the chart to find the amino acids. Okay, so you had 3 of those to do. Then you were supposed to, yesterday's assignment, or for you all it was Monday, was to did I do this with you all? Did you all start this? No. Okay. Okay. So this is new. This is new. All right. So this is the assignment. Yeah, I pulled it up. We were going to get to it, but we didn't quite get to it. All right. So what this one does is we're looking at mutation. So this is our mutation page. In particular, we are making the protein hemoglobin. And hemoglobin, and most of you have heard of it. If you ever been to the doctor, they looking at your hemoglobin count. That is your red blood cells. So we are looking at what a normal code for hemoglobin would be and an abnormal code for hemoglobin. So that's what this exercise is. And so you're going to use the chart, and this one just happens to have the circular one on it, but use whichever one you're most comfortable with. And you're going to create a row of RNA. Then you're going to create, based on the row of RNA, and this one is already divided into a triplet code for you. You're going to use that code that you created. For example, with the first one, the C is going to code for G. The A, because it's RNA that you're making, will code for U. And C again will code for G. So you're going to look on the chart for C. So again, start from the middle, work your way out. So you want the wedge that has the blue C. And the next one is U. So you want that kind of orange looking box. And then you want to go, what did I say? CUG. Sorry, G. I'm sorry. This is G. GUC. Because I knew I was ending up in the wrong place. Okay, so let's start with the G. All right, so we're doing G. G. Starting the yellow one up at the top. And then we're going U again, which is still orange. And then we're going to go out to our third circle. And again, we have 4 different letters for our third circle, but all of them will lead to valine. So valine is going to be your first amino acid. So you're going to do that whole row. And the first row is what the DNA should look like if it's normal. Second row, you're going to notice that there's one change, and you're going to create one different amino acid. All right. So you're going to do the same thing up there. Then you're going to answer these, it's really 2 questions and a paragraph that you have to fill in the blank. All right, this one, I'm going to help you with this one. It wants you to, because I had the other class, I got on them because they AI'd the answer, and that's not what you need to do. You need to show that you understand that DNA has the directions to make the RNA, and the RNA is read in sets of 3, which makes the codon. And the codon codes for an amino acid, which makes a protein. So you need to relate these words to each other. DNA makes the RNA. The RNA has the bases, which make the triplet bases, which make the codon, and the codon then gets the amino acid that makes the protein. That's what number 1 wants you to say. Okay, so say that some kind of way, but try not to copy and paste an answer, because it's not going to say it the way you need it to be said. All right, what you have in the picture over here are the circular ones are what a red blood cell should look like. And the other one, we're looking at the disease sickle cell anemia. So we call this shape of this a sickle shape. It's not round, it's kind of a curved, but it's still a red blood cell. So a good red blood cell's function is to carry oxygen. So if that's its function, when it is properly made, it's doing it well. It carries a lot of oxygen. When it's not made well, it doesn't carry as much oxygen. So the next question asks you, how does the shape of the protein relate to its function? So that's what you want to say about that. You want to be specific to this case. In this case, you want to talk about what is the difference between the function of a normal red blood cell and the function of a sickle cell shaped red blood cell. Okay, after that, you want to just fill in the blanks. This is not multiple choice. That's there to let you know that there are 4 somethings. So you want the word, what do you call these 4 things? That's what word goes in the space. And the rest of these, you just want to make sure that you are telling the correct story as far as how to make a protein. And then these last 2 are specific to this particular activity. All right, so I'm going to stop talking so that you get a head start on working on this, but this was today's assignment. Any questions? Anybody need me to re-explain a portion of this? Are we good? Everybody was able to download the worksheet? All right, so for you, this is due tomorrow. All right, please ask questions if there's something you don't understand. I did put that video in the playlist, so if you need to go back and see how to read the codon chart, it is there for you. All All right, I stopped sharing because I need to do attendance. Look at there, everybody's here today. That's a good thing. That's a good thing. All right, so the assignment for today is to do the mutation activity. And that is due tomorrow. All right, so while you're doing that, I will look at things that are graded. Again, anything that is very late, I may take the Canvas late off of it, but it's still going to be graded late. But late is better than zero. I do have to, before the week is out, send letters home. So anybody who's not up to date, expect a letter. Again, if you have a question on how to do the worksheet, how to read the code, please ask. All right, some of y'all are falling more behind because you're spending more time looking at your grade than doing the grade. All right, so by tomorrow we want to finish this unit. We only have one more section to do before we're done, and that's technology. And with the technology, we're going to go back and look at your Halloween DNA question, because a lot of that question had technology in it. Okay, folks, it is time to go. So make sure I have somebody for my next class already coming in. So you all work on that. Make sure when you come in tomorrow that you're working on it. And